I normally say it's 5 o'clock in Copenhagen, so it's time to get started, everyone. This is our 11th webinar, and today I am really honored. I have Jeff Bland with me, with us, because it's not just me. If you weren't here, everyone, this wouldn't happen. So I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, a group of companies that are dedicated to help changing healthcare through functional medicine and personalized medicine. And remember, lab tests, supplements, log onto your practitioner profile, you can get it all there. So, and you can always find links in our mailings and so on. We are recording, I hope. Let me just double check. Yes, recording. So the recording will be shared as soon as we can. And then I just want to say before, and just hang on to the end as well, because I'm going to say something at the end as well. But DNA Life have created a partnership with PLMI, the Virtual Thought Leaders Consortium, in October 2020. And even though it's been a COVID year and normally conferences are on site, they have made it a goal to create the event online and then thereby creating it, making it global. I love the PLMI conferences. I've been there to them myself personally, and they are amazing, and they're also thought-provoking and very foresighted. We will bring a special offer for the October event for all you who's online today, and I would say it's a very good investment to signing up for that. Of course, and perhaps the best investment apart from getting a DNA Life test. Anyway, stay tuned for the offer at the end when Jeff has finished his presentation. So, Dr. Bland, Jeff, I'm Danish, so we're always on first name. I probably don't need to say very much because everybody knows you. You're a star within our field. You are Mr. Functional Medicine, you're Mr. Personalized Lifestyle Medicine, and you're also Mr. Systems Biology. You might be Mr. something else as well, but you're probably one of the most foresighted people within our field that I know of. And you're very warm and kind. I feel very comfortable in Jeff's company. So, and I was with Jeff and Susan, his lovely wife, in Hong Kong just over a year ago. Thank you to Javid and his family. I think Javid is online as well. So, and that's when we kind of loosened up a little bit and danced in snakes and sang hokey cokey and whatever it's called. And we also had the pleasure of eating traditional Chinese food with chicken and duck feet. But that's when Jeff and I decided to start talking about fasting mimicking diet. How good that is. I think we actually competed a little bit about how many times we had done it. So, but enough about my talk. I hope these webinars is able to create a family-friendly feeling, and that you as practitioners out there in the world feel united but apart. So enough from me. Jeff, I will pass the stage on to you, and you're welcome to share your screen, and I will take questions. So if there's anything you can write it, I will try, and you know, Jeff it goes very quickly. I will try. I have a brain that's maybe not working quite as quick as Jeff's, but with Jeff in the end. So stay tuned. Jeff, your go.
Well, Anne-Catherine, thank you. And boy, oh boy, do I feel privileged to be with this extraordinary group of people all over the globe. It's pretty amazing, this technology. And on one side, we feel kind of isolated with COVID-19 and kind of the stay-at-home situation that many of us have been experiencing. But on another way, we've learned how to use this technology to our betterment to really create alliances and cooperation and linkages, and particularly now that I can see so many of you on the screen, it really makes us recognize that we do live in a world of community, and that we are aligned and connected, and that we are forging ahead to hopefully create a world with less disease and more function and more joy. And I think that's all of our shared goal and aspiration. So it's a real privilege to be here with you all this morning, and it's morning here. I'm in the Seattle, Washington state area over in the west coast of the United States, so it's morning here. But I'd like to share some thoughts with you all about this thing that we came up with that we call functional medicine in 1990. And when I look back at this, I recognize that the development of new ideas, of new concepts, of innovation, often, and maybe even more often, doesn't occur through a well-planned, orchestrated type of sequence of events. It occurs spontaneous through a group of people that kind of collaborate, and somehow an idea pops up and it gets sticky, and people dedicate some of their life to that idea because they think it's interesting or important. And then if it really has merit or value over time, it grows up and it gets enriched and it gets more flavor and it gets more people bringing their brilliance and their concepts into it. And that's what's really happened to this concept of functional medicine that was really born out of these meetings that my wife Susan and I had hosted in Vancouver, British Columbia, on Vancouver Island, actually, in Victoria, back in 1989 and 90. And out of that group of about 35 people that we had invited to attend these meetings, emerged this concept of functional medicine, which I am just quite amazed, actually. If I think about it, it's 30 years, 1990 to 2020, that's 30 years. And now over 220,000 practitioners have gone through its courses, and we're in countries all around the world with this concept. And if you would ask me, would I ever believe that this would happen? Sitting there in 1990, I would have said, no way. You know, it just seemed like a small group of people that had this interest in trying to find a better way to improve people's health. And we started, you know, really exploring this idea. And then, you know, once it got into people's minds, it's like a virus. it kind of replicates in our minds, and it starts growing and it becomes its own. And it's really exciting to see that, how that's happened. So if I could just share with you some thoughts, we were thinking we were pretty smart that we came up with this idea. And it was about 10 or 15 years after we started, so around 2005, that one of our doctors came to me and he said, so Jeff, do you realize that there is a paper in the 1871 issue of the Lancet on July the 1st? Think of that, July the 1st, 1871 was an article by Willoughby Wade, who was the dean of the medical school at Birmingham in the United Kingdom, in which he wrote this article and gave these lectures called Clinical Lectures on Functional Medicine. Yes, 1871. And so we didn't feel so smart after we heard about this, that this had actually been around. And what did he say? So he said some really interesting things that were very prescient that we thought we kind of had invented. He said, so what is a disease? Now remember, this was written in 1871. So what is disease? In old times, people used to think that a disease was some actual entity or thing which had gotten into the body in some way and was there laying hidden and secreted and was to be cast out. This idea, which we now know to be true only a few specific instances, was at one time the general thought. The conclusion today, however, is that all disease is disordered function. Again, that was written in 1871. And then he went on to say, here then I say, is the highest justification for all treatment being based upon the principle of restoring disordered function to order. And this is what I have ventured to term functional medicine. Well, shame on us. I probably should have known about this article. I consider myself a bibliophile, but I didn't know about it. So, you know, we kind of co-invented, but he certainly preceded us by 100 years. So what is the functional medicine epistemology? I mean, what makes it a system worth thinking about? And I think that there are really a couple of independent issues that uniquely define functional medicine. It's built around the construct that we are all born with our unique genetic architecture. I call that our book of life. And our book of life is encoded in 23 chapters, half of which is written by our biological mother and the other half is our biological father. And we all studied somewhere in our schooling the concept of genetics as a Mendelian concept. So we got this concept that there are dominant characteristics in our genes and there are recessive characteristics in our genes. And I think for many of us, certainly I would say for me, as a student growing up all the years through schooling and into medical school, I kind of felt that this was a deterministic model of our health, that it kind of locked everything the day we were born. We didn't fill out an application card. We just got the luck of the draw. And some people maybe got a good luck of the draw with their book of life, and other people wasn't so fortunate, and they had not as good a luck of draw. And that was the model that really got me into medical school, probably, was the ability to recite that kind of a concept on tests and exams and understand genetics from that perspective. But it really had never, for me, fully described why it is that certain people get diseases and others do not, even within the same family. And so I always felt that there had to be another component of this. And of course, the other component that we now know very clearly is lifestyle, environment, diet, exposure to various substances. That's the stuff that works on our genes in terms of how our genes are going to be expressed to give rise to our phenotype. So the functional medicine model is a different model than this deterministic model of the Mendelian genetic underpinning. It does recognize, certainly, Mendel's laws are true, but there's another factor which we say genes plus environment plus lifestyle equals phenotype. And that's how a person looks, acts, and feels. We cannot, at least today, change our genes, but we certainly can change the modifying factors of environment, lifestyle, diet, and so forth to modulate then how our genes are expressed. And that's a very important, different concept than the way that I kind of was educated to see that we have that plasticity, we have that capability. So genes plus environment plus lifestyle plus diet equals the phenotype, and all those things in red are modifiable. That's where the action point is. Well, then we say, well, what are the targets then? If you're looking at a patient or an individual, not necessarily a patient, looking at an individual, what are the various functions that are reflective in how their genes and environment are matching? In other words, when you, as a practitioner, if you see them in that primacy of an exam room, what are the things that you're looking at that are the phenotypic outcome of how those people's genes and environment went together? And that particular question was really a question that we, as the founding members of the Institute for Functional Medicine back in 1990, started to think about. Could we break down the various subsets of activities that an individual would be exposed to that would then translate into how it would be seen in the signs and symptoms in their body? And when we did that, we came up with the following categories. Now, these are not locked in stone. These were not passed down on the mount on a tablet to us. But after a lot of consideration, thinking, and looking at the literature, this is how we eventually broke down into compartments the various aspects that we felt built together to give rise to the ultimate phenotype of the individual. And that included what we called assimilation, which is the gastrointestinal process of breaking down big stuff to small stuff and absorbing it. So proteins get broken down to amino acids, carbohydrates get broken down to monosaccharides, and fats get broken down to fatty acids, and how that all then gets absorbed and utilized and then ultimately taken up so that we're more than we eat. We're what we eat and how it gets broken down, absorbed, transported, taken up by cells, metabolized, and excreted. That's kind of the process that we thought about. So that was number one. Number two was our immune defense, which we call defense and repair. All the processes that relate to how the body defends itself against internal and external foreigners or potential toxic or noxious agents, and then how the body repairs itself, like DNA repair mechanisms and cellular repair mechanisms. Next, we talked about energetics, bioenergetics, and we were, I think, maybe the first group in our field to really start focusing in on mitochondrial bioenergetics. And I'm fairly proud of this because the first seminars I did for doctors on mitochondria was in the 1985 period. And so I think we were quite a ways ahead of the pack and leading in an understanding of how bioenergetics play roles in different tissues in regulating their function, and what are the factors that modulate or modify mitochondrial bioenergetics. And then the next was biotransformation. And we asked the question, how is it that the body that's exposed to all these things that we'll experience in the course of a life, not accumulate that so it ultimately gums up the work and causes disease? It has to find a way to get rid of the inevitable byproducts of its own metabolism or the toxins that it's exposed to, the so-called xenobiotics, big X word. And so that has to do with, obviously, the process of detoxification, biotransformation. And it's interesting to note that the first detoxification enzyme, so-called cytochrome P450, was discovered in late 1960s, early 1970s. So when you really think about it, that's a fairly recent discovery in human physiology, how we actually biotransform, get rid of, and excrete substances so that they don't accumulate in our body and produce toxicity. And then we learned, obviously, that there are various nutrients like glucosinolates that are found in cruciferous vegetables that actually can speak to the genes that regulate the biotransformation process and can upregulate and can activate or modulate that activity. So the diet can play a role in the biotransformation process. So I'm kind of proud that we were one of the first groups to really put that model together as part of our curriculum. The next is communication, intracellular communication. And this is a very, very big question. How does something that happens on one part of the body affect somewhere else in the body? So you say, well, we have the nervous system, so that's one important part of the communication system. But there are also chemical communication systems. We now know about the endocannabinoids. There's something that's topical right now. The endogenous cannabinoids that are involved with intracellular communication, which regulate the activity of a specific set of receptors that are members of the family called the G protein coupled receptors, which we now recognize sit on the surface of virtually every cell type, and that there are over 150 different types of GPCRs with different ligands that bind to them that activate them in different ways. And those can all be modified and modulated by specific aspects of our lifestyle, environment, and diet. So here's another important part of the function of our body that can be regulated if we understand it and we can identify it. Next is transport. And so how do things get carried from one part of the body? Like how does glucose as a fuel get from the gut ultimately inside a cell where it can then be used as a source of energy? And so now we say, well, there's all sorts of mediating substances that help in transport. There may be binding proteins. There might be regulatory hormones, as in the case of glucose, it would be things like insulin and glucagon. So we start to say, gee whiz, could there be breakdowns and could there be dysfunctions in these transport processes that give rise to specific types of dysfunctions that later we call a disease? But it's really not a disease, it's a dysfunction associated with this process, and that process is regulated by certain aspects of a person's lifestyle. And then next is structural. Obviously, we think of that skeleton at over in the corner of our anatomy lab, and it looks like just a collection of calcium hydroxyapatite deposits that are in an interesting geometry we call the skeleton. But actually, as we know, the skeleton is an extraordinarily important bone, an important dynamic part of our body, because every blood cell, be it either white or blood or red, comes from our bone marrow. So our structural system is directly connected to our metabolic system. And now we know there's a gut connection to the bone, there's a bone connection to glucose relationships, there's a bone connection to adipocytes, and so you can have fatty bone. And so all sorts of interesting new things that we've learned about structure and how structure, like exercise and activity, putting stress on your skeletal mass, then increases its density. So that brings the whole concept of movement and physical medicine into our story as well. And then lastly, of course, and certainly not the least, is the psychospiritual component, this non-quantifiable energetic component of our body that has a superbly important role in overseeing, regulating, and modulating function at every level around all these processes. So this is how we then undertook the functional medicine model, was to use these targets. And clearly all of those are tied to our genes because different genetic characteristics may regulate in different ways how those function. So this was the basis of the functional medicine model in the formalism as we started to build it out in the 1990s. And we built it back very strongly on the shoulders of what I consider some giants, people that I have had the privilege of knowing and working for, and now deceased, but certainly were remarkable. Roger Williams being one, the head of the biochemistry department at the University of Texas for many years, the discoverer of pantothenic acid and folic acid and vitamin B6, pyridoxine, a really an incredible nutritional scientist, and who developed really in 1949, this article was first published in the LANCET entitled Biochemical Individuality. And he actually developed a concept called the genetotrophic disease concept back in 1949, in which he said that there are many diseases that are caused by the insufficiency of specific nutrients. I want to emphasize insufficiency, not deficiency. So these are not necessarily people that have scurvy, beriberi, pellagra, xerophthalmia, rickets, kwashiorkor, or marasmus, the known nutritional deficiency diseases. These are people that have insufficiencies of specific nutrients that translate then into dysfunctions that we then call a disease. And he called these genetotrophic diseases because it's partly related to their genetic uniqueness and need for those nutrients, and then partly related to the origin of that disease through the insufficiency of those nutrients. So it's a genetic relationship. And what he said is we shouldn't put standards like the MDR or the RDA for nutrient needs, because those are not able to tell you what an individual person needs. There's a greater variability from person to person when you start looking at individual nutrient needs to support function than there is for nutrients just to prevent in a population the presence of scurvy or beriberi. So that was a very important concept that we built into our functional medicine model. And in fact, this is one of Roger Williams' early papers that really discusses this genetotrophic disease. This is the LANCET paper in 1949. So we derived a lot of our thinking from these great thought leaders. And another one of those great thought leaders, more recent, and a gentleman that's still at 92 years of age, still writing papers in the scientific literature, is Bruce Ames. Bruce Ames was for years the head of the biochemistry department at the University of California at Berkeley. He's also the developer of the Ames test. Some of you are familiar with that. It's the most common test used for assessing the potential carcinogenicity of chemicals. Remarkable thought leader. I could go on and on to talk about the contributions he's made, but let me just speak to this one paper because I think it exemplifies the nature of his contributions and how we bake this into the functional medicine model. So this title of this paper probably looks a little bit esoteric. High dose vitamin therapy stimulates variant enzymes with decreased coenzyme binding affinity, increased Michaelis constant. Its relevance to genetic disease and polymorphisms. Okay, that's the title. It seems like you need about 3 references just for the title. It's pretty sophisticated. So let me say what I think this article tells us. He and 2 of his postdoctoral students did a tour de force bit of research. It was literature research. And this paper has, I believe, over 400 references. And what they did is they went and they looked at every paper they could find, scientifically valid paper, that described the role of a vitamin in modulating a biochemical function at the cellular level, and asked the question in terms of each of those vitamin factors, what impact did it have upon that enzyme, and how was that enzyme regulated by its own genetic polymorphism, knowing that these particular enzymes are proteins, and that we know that proteins come from genes, and that we know that genes may have different polymorphisms to produce the same protein. Are we okay so far? Have I lost anybody on this? So if the genes produce the proteins, but the genes are slightly different by one letter changes that we call SNPs, then could it be that there's a variant need for specific nutrients to affect the function of specific enzymes in specific cells that regulate specific function in our body? I hope you got that. That was, I know, a lot of stuff put together there. And of course, the answer to the question is yes, there are a lot of variation. And what he found was that there were certain kinds of SNPs, certain kinds of variations that may require up to 50 times or more the level of a specific vitamin-derived cofactor to make that enzyme work properly over the amount that it would take a person that has the wild type or normal genetic structure. Let me say that again. I think that's an important point. They found that there was evidence that there, in a number of cases, were genes that were polymorphic so that the proteins and the enzymes that they made to be active would require 50 times the amount or more, 50 times or more of a specific vitamin-derived cofactor in order for them to function normally. So that means if these people were eating a diet that was recommended based upon normal levels of intake of vitamins, for them, it would be insufficient to meet the needs of those genes to regulate that function, and they would have suboptimal function, which over time would produce a suboptimal genetotrophic disease, to go back to Roger Williams' concept. So this is the basis for what Dr. Linus Pauling called orthomolecular medicine. In 1968, he wrote his classic article in Science magazine called Orthomolecular Psychiatry, in which he described why certain psychiatric functions could require much higher doses of vitamins to prevent something like schizophrenia. Now, where did he get that model from? He collaborated with Dr. Abram Hoffer, a psychiatrist and also a Ph.D. in nutrition from Canada, to regulate psychiatric conditions with megavitamin therapy. And actually, Dr. Hoffer was well known. People came from all over the world. He just passed away a few years ago at 96 years of age. In fact, he was seeing patients the day before he died. I was very fortunate to interview him about a month before he passed away. But Dr. Hoffer came up with the concept because he grew up as a farmer's boy in Saskatchewan, Canada, living on a farm. And he recognized that in the animals that they had on the farm, that there were certain animals, if they didn't get higher levels of nutrients, that they produced this strange behavior. So as he got into psychiatry as a medical student, and he also got a PhD in nutrition, he started saying, well, hold up, what is the sign of the deficiency of niacin, vitamin B3? You would say, well, that produces a problem that has the 3 Ds. That's the 3 Ds is the way you learn about the deficiency of niacin. And what are the 3 Ds? Dermatitis, that's 1 D. Diarrhea, that's a 2nd D. And what's the 3rd D? Dementia. And the form of dementia that's often seen with vitamin B3 deficiency, we call that disorder pellagra, by the way, resembles schizophrenia. So he wondered, could it be that certain schizophrenic patients are really pre-pellagrous? They're not, functionally vitamin B3 deficient, but they're insufficient. So that whole model, the interworking of the Linus Pauling work with the Abram Hoffer work, with the Roger Williams work, really, I think, was the ground from which was born the field of nutrigenomics. That was the precursor to the field of nutrigenomics, which came later in the 20th, 21st century. Now, how do we know all this is true, that I'm not just making it up as I go along? I think the best way to know this is true is to take monozygotic twins and to study them, because they share the exact same genes, but yet as they age, unless they're living in the same household, monozygotic twins in their phenotype become less similar. And now we have seen over the last 20 years, because we now have gene sequencing capability, we have seen published more than 1,000 studies on the nutrient differences of monozygotic twins, the differences they have in their phenotype. So how do you account for that if they have identical genes? It's because something has happened to the way their genes are expressed over time from the environment in which they live. And this is just one example of looking at 3-year-old individuals, and these are monozygotic twins, and then 50-year-old individuals. And you notice by the height of the dark versus the light bars that they become less similar over time. There's more dissimilarity on the bars of the right, which are the 50-year-old monozygotic twins, than when they were 3 years old. So I hope you can see that from the bar graphs. So this is a very interesting case as to why, if they have the same genes, do they have these different characteristics? It's because their genes have undergone epigenetic changes through methylation and other things that occur from their lifestyles that have happened. So the genes in themselves are the same, but the way the genes are expressed become different based upon lifestyle. And so this is the paper that appeared in the Proceedings of the National Academy of Sciences that was kind of a landmark paper. Epigenetic differences arise during the lifetime of monozygotic twins. That was the title of the paper. So when we put all this together, we recognize that the biology of health is different than the biology of disease. Let me say that again. The biology of health is different than the biology of disease. If you want to increase health span, you need to ask a different set of questions about the function of the individual than you do about the biology of disease, in which we as people in the health sciences have been trained that the absence of disease is the presence of health. That is not true. The absence of disease in and of itself is not a definition of health. The absence of disease stands alone as the absence of disease. The presence of health has to do with the function of the body at all of these various levels of systems that I just talked about. And that's what this article that was just published in Cell, it's a landmark paper, just appeared out of work at the SOC on the mechanisms of health distinct from the mechanisms of disease pathogenesis. And I think it's a very interesting new chapter in how we're seeing function. So the biology of health and the physiology of health really talks to how we have resilience. And I won't spend a lot of time on this, but this article does a really nice job of going through disease tolerance, which I call resilience, and functional reserve, and how that relates to all these various, again, what they break down as systems of the body. If you look at these, however, just look at the ones they talked about: growth and development, socialization, energy balance, detoxification, acid-base balance, oxygenation, osmoregulation, energy balance of body composition, thermoregulation, and macro and micronutrient physiology. Does that at all sound like functional medicine? Yeah, it's another way of reframing what we've been talking about for the last 30 years. So what happens if you put the functional medicine concept to work in a group of individuals? And this is collaboration that we did with the Institute for Systems Biology, Dr. Lee Hood and Dr. Nathan Price in Seattle. This is a world-renowned research group. And they did multi-omic biological age determination, looking at the connection between all of these types of things. That would be genomics, proteomics, metabolomics, microbiomics, phenomics, wearable devices, I was one of these, what they call pioneers. I was one of the participants in this study. There were about 350 individuals that were followed for 3 years with literally I had over 1 million data points collected on me over these years. And then a very high level analysis, statistical analysis was done using informatics and artificial intelligence, and trying to look at the clustering of the data as to what constitutes health, not just what constitutes the absence of disease. I'm just going to summarize. This is just one little tiny bit of the data set from this study, by the way, which was published in this paper last year. I'm just giving it the tip of the iceberg, but the reason I chose this is when you rank order the characteristics that were most associated with high function, they were related to immune system function. And they were related to things like the neutrophil lymphocyte ratio. They were related to how your body's immune system is functioning and if it's in a state of chronic inflammation. We call that, as you probably know, inflammaging is the term that's been associated with it, inflammaging. And so the connection of the genes to the connection of the biomarkers that are associated with aging gives you some understanding of the architecture of health of the individual. And the more shifted that pattern is towards inflammation, the more disturbed the physiology is and the lower the health reserve. This, by the way, I think relates a little bit to what we're seeing with COVID-19 now, with individuals who have the most serious sequelae when they've been infected. Those are people that have already the inherent disturbance of their metabolism through these imbalances of their immune system. Before they ever even got exposed to the COVID-19 virus, they were in a state of preparedness for greater infection risk because of their disturbed physiology. So we would say they were functionally imbalanced, even though they were not disturbed. Some people call those biomarkers, or call them comorbidities. I think comorbidities is a little off-putting term. I don't think it's actually the right term. I call these dysfunctions that precede or set the tone for increased risk to infection. By the way, those are all modifiable. So we can reduce the incidence of serious infection of COVID-19 by treating what I call the pandemic within a pandemic. I'm just publishing an article today in Medium entitled COVID-19, the Pandemic Within a Pandemic. And the pandemic in which COVID-19 finds itself is a pandemic of metabolic syndrome. 14 million people die globally every year from the sequelae of events that are associated with metabolic syndrome. It is far more prevalent than the people that are dying from COVID-19. But COVID-19, when nested within metabolic syndrome, makes it much more virulent and much more pathogenic. So when I put all this together and asked, did this really translate all these words that I've been sharing with you and all this kind of stuff, does it translate into people actually getting better? If you were to apply this model, the functional medicine concept that I've been describing, is it able to actually deliver better patient outcome by this kind of strategic thinking in patients that have what I call walking wounded chronic illness? I use the term walking wounded because often these are the people with shuffling feet and stooped shoulders who go through life saying, when am I going to get sick enough that someone knows what's wrong with me? And they may be on multiple medications to treat symptoms, but they're not getting better. They're feeling ill. And we call these, at least I call these, they're not really diseases, although we might give them a name for reimbursement, but they really are syndromes. And if you think of the number of syndromes, chronic fatigue syndrome, fibromyalgia syndrome, cardiac insufficiency syndrome, hypertensive syndrome, I mean, I could go down the list. These syndromes are really what people that have as chronic health problems come to the offices of health providers, depressive syndrome, to have managed. So how do we know that the functional medicine model, when applied to these conditions, are as successful or better than standard of care, which is make a diagnosis and give a drug, basically. So I'm very excited, and some of you already know, that we have a Center for Functional Medicine at the Cleveland Clinic in Cleveland, Ohio, which is arguably one of the top medical clinics in the United States. It's a huge medical center, over 40,000 employees in the Cleveland Clinic, so it's an enormous, large facility. And they have obviously a general family practice clinic there, which does very good work, the standard of care. But we were very lucky that the CEO at the time of Cleveland Clinic, Toby Cosgrove, a very forward-thinking, innovative cardiovascular surgeon, actually, who is the was the CEO of Cleveland Clinic, really was struck by the functional medicine model, and particularly struck by the way that Dr. Mark Hyman described it. And so that led a discussion between Mark Hyman, Institute of Functional Medicine, and Toby Cosgrove that eventually led then into Cleveland Clinic opening its doors for a Center for Functional Medicine. That was about 4 years ago. One of the requirements to set up that center was that we would be engaged in doing outcome studies to demonstrate that the functional medicine model was valuable. So this was our opportunity to do a proof of principle study. That has been ongoing, doing comparative analysis of patients with similar symptom scores, going through the traditional standard of care family medicine practice in the Cleveland Clinic, and similar patients going through the functional medicine center in the Cleveland Clinic. So it's kind of a split study with people getting the standards of each one of those models. The outcome of that study was just published of 7,252 patients going through the Functional Medicine Center and 1,595 patients going to the Cleveland Clinic Family Medical Center. And what was found in doing a comparative head-to-head analysis was that both of the models worked. Patients got better, but the functional medicine model had a statistically significant improvement over the traditional medical model. It led to all patient-reported outcomes improving as contrasted to the traditional standard of care model for family practice. And this was published, as some of you know, in October of 2019 in JAMA, in the Journal of the American Medical Association. So we're very excited that we have our first peer-reviewed kind of collaborative study doing a comparative analysis of outcome of the functional medicine model versus the traditional standard of care model for the management of what I call garden variety walking wounded patients in a major medical center. So this is not the last study, this is the first, obviously, but I think it's very encouraging to demonstrate that this model that we started some 30 years ago really does deliver improved patient outcome. So that then leads us really to how does this relate to today and this challenge that we're experiencing through COVID-19. And I wrote this article that appeared in Medium a few months ago, which it got quite a bit of controversy here in the States, called COVID-19, the quintessential example of a planetary lifestyle disease. Because if you think about it, as I said earlier, the pathogenicity of the condition COVID-19 is really related to lifestyle. We see it most serious in people that have disadvantaged medical services, people that are a lower socioeconomic capability, people that are impoverished on many different levels, poor nutrition, poor social structure. And so we start recognizing that there's something that connects infectivity of this virus with the social determinants of disease and the nature of their function, which is really the perfect model for functional medicine. So if we look at what's learned about COVID-19 really quickly, we learned, and this was a very interesting seminar I went to recently with looking at monitoring the immune system and its relationship to COVID-19. And just to say it simply, why do certain patients experience severe disease and others really are almost asymptomatic? Why are younger children seemingly spared and have very infrequent disease, whereas others that are older have very serious disease. And what role does the immune system play in this? And when we start looking at it, we see that a lot of this has to do with the function of the innate immune system and its interrelationship with the adaptive or acquired immune system's activity. It has to do with the activity of these cells that make up these immune system components. And so if we were to summarize what we've learned to date about the immune system's function and its connection to COVID-19 serious infection, here are the summary. T cell counts may be preferred to total lymphocyte counts because total T cell tells us something about the activation of the immune inflammation of the system prior to the infection. Levels of decrease predicts the moderate to severe disease, so we get lymphopenia because you actually start to have an increased number of the immune cells that are engaged in inflammation and a lower that are involved in the actual first level defense. And so by characterizing immune cell function, we can really understand something about the infectivity of COVID-19 as being associated with a dramatically confused immune system. The people that have the most serious problems are those that have this precedent confusion in their immune system, which we call inflammation, in a state of inflammation. And those are the people that go on to have the highest risk to the cytokine storm. And the fact that it looks like SARS-CoV-19 serious infection is what is called viral sepsis. It has like a septic kind of relationship that activates and feeds forward the inflammatory cycle in an individual that is already in a presaged inflammation status because of the nature of their functional imbalances. So this is kind of how in this article they talk about the first level of immune activation occurring on the mucosal surface of the upper component of the respiratory system. So here we talk about mucosal immunity. So we see also, as you know, mucosal immunity in the gut is associated with COVID-19 as well. So all of your immune surfaces and immune cells that are populating those surfaces are in a presaged inflammatory state. So that then takes us to why would we have a imbalanced immune system to start off with? And it takes us back to some of the functional medicine principles like toxicity. And as we know, we live in a world where we're being exposed more and more to persistent organic pollutants and things that modulate our immune system in such a way to put us on guard all the time. And if we look at the World Health Organization public impact of chemicals, it's actually staggering to read this report and to see how significant inherent toxicity is becoming in all of the world in terms of its established altered function in individuals that makes them more vulnerable to all of these chronic age-related diseases, how their genes get turned on, the cassette of genes that are related to inflammation. And so we have a new field that is emerging called toxico-epigenomics. How do the toxicants that we're exposed to influence the exposure and expression of our genes, so that environmental chemicals and their impact upon the epigenome and its regulation of our function, which then makes this concept of biotransformation and detoxification ever more important, and gut restoration ever more important. So exposures lead to molecular changes in our epigenome, lead to cellular changes in our function, lead to tissue level changes that then produce individual health effects, which then ultimately aggregate together to become population health effects. But they start with individuals and how their own individual genome is influenced by the exposure to these substances. This is a functional medicine problem. So if I could just kind of close, and thank you for being so patient with me and allowing me to wax here on and on philosophically, but I think we're at the threshold of an incredible revolution in thinking about health and disease. And I think that the immune system is going to be the entree point for many people to start to understand how to measure and understand how our genes get turned on and off into our function and how they regulate ultimately our risk to these major disease categories. So we're, as Anne-Catherine said initially, we're putting our 8th annual Thought Leaders Consortium with PLMI as a virtual meeting this year. I guess the good news is it makes it more accessible to everybody in the world that doesn't have to travel to the States, but it's going to be focused on immune rejuvenation, this personalized approach towards medicine in a pandemic era. And I think it really builds off this functional medicine model, asking what do we need to test? How do we use our genomic information? How do we then translate that into specific personalized intervention therapies that then deliver the best of functional medicine to people in need? So I hope you all consider joining us on virtually on the 9th and 10th of October. And I want to thank you so much for the kind privilege to share with you some thoughts and ideas. I hope a few things might be helpful for you. And Catherine, I'll turn it back to you.
Excellent, Jeff. You are, as usual, blowing our minds. And it's really nice to have the background and the story and a really thorough way of explaining the functional medicine and leaning up to what we are standing here with today with the COVID-19 pandemic. And now you're taking on immune rejuvenation as a topic at the PLMI. I'm really proud again, DNA Life for sponsoring with PLMI. And we will, in the recording in the next few days, share the link so that you can sign up for PLMI. And our group, so you guys, our community, our friends, and so on, will get a really good discounted offer for these. So the normal price is $250, and you will get it. We will get a shared discount code with you, so you will get it at a half price. And for those who signs up for PLMI, DNA Life is also offering, because there's a lot of focus on genetics, and we want to understand the genetics. That's kind of like our foundation. And then we have the environment, as Jeff elegantly has demonstrated today. So we will offer, of course, a great price on the DNA Life tests. So what we will do is that we will offer DNA Health. For those of you, you might know it already, but that's kind of our base DNA test that looks into genes in relation to inflammation, oxidative stress, methylation, etc., etc. We will offer that at $99, and then you can add each other. We have one focusing on estrogen, we have one on weight management, we have one on sport, we have one on mind in regards to dementia, in regards to addiction and mood disorders as well. So we have 6 tests in total in the DNA Life package in nutrigenomic testing. And if you buy all 6, you get it at $199. And I said, so how much do you save? You save quite a lot, because its normal price is over $600. So that's for those who sign up, and there will be we will explain it all in the mail, so that it should be pretty self-explanatory once you go in and sign up and so on. But it's for those who sign up for the PLMI, we have this special offer, and you can use it for yourself or a family member, and we allow for you to buy a maximum 2 at that offer. So now you had to listen to that as well, Jeff. But anyway, I hope you survive. So thank you for giving us background. Thank you for giving us vision. I love it. I know everyone else loves it. There were words like, it's music to my ears. You're a well of inspiration. Thank you for all your pioneering work. People love you, Jeff. And thank you for sharing your morning with us. And well, hopefully we will see you at the PLMI event. I'm sure you will have something to say there as well. Yeah,
I'm doing something I'm very excited about. I'm going to have, because it's virtual, I'm going to offer people, if they want, an hour with Jeff Bland during the program, where they can just Zoom with me directly for an hour to discuss what they're learning from the meeting, which I'm going to learn a lot from that. Yeah. So it'll be fun just to have a group chat during the meeting to gain opinions and give feedback.
Wow, I'm looking forward to see what you have to offer, because that sounds really exciting as well. So thank you. Yeah, anyway, recordings will be sent out, follow-up emails will be sent out. Some are asking if you would mind sharing the slides, then you can just send them to me if you wish to, and if not, that's also okay. We will share the recording. So
No, I will definitely, I'll email as soon as we get off this call.
Excellent, thank you. So to everyone, thank you for staying tuned, and hopefully we'll see you at another webinar that I will get going within hopefully the next few weeks. And then virtual hug to you all, and you, Jeff, and so on. So take care, everyone. Thank you. Thank you. Bye.
Be well, everyone. Bye-bye.